I've spent all this course so far talking about how viruses get to mRNA. Today we're going to figure out what they do with it. And of course, this is all about translation or protein synthesis. Now, viruses all need to make mRNA that is readable by ribosomes. So, because no virus encodes a complete translational system, they are totally parasites of the host translational system. So you may ask, why are you teaching us a lecture on translation if that's a cellular process? Well, if that were the case, yeah, I could argue that it wouldn't be part of a virology course. But in fact, viruses will modulate translation in a variety of ways that benefit their replication cycles and take away from the host. It's all about competition. So that's what we're going to mainly talk about today, how viruses manipulate the cell's translation system so that more viral proteins are made. Because after all, that's their goal. They want to make a lot of proteins and make a lot of virus particles. And we're going to start by just having an overview of translation, because I don't know how much you know about it. Someone remarked last week that they thought ribosomes made DNA, and that the synthesis of protein is called transcription, both of which I hope you recognize are not correct. So I want to talk a little bit about translation first. So translation is the art of protein synthesis, making proteins using mRNA as a template. We'll talk about that mRNA in the middle, which is made from all of the 7 different classes of viruses that we've talked about so far in this course. So let's talk a little bit about the mRNA itself. We've already had an introduction to this when we talked about transcription and RNA processing. Here on the upper right is a typical messenger RNA, a cellular messenger RNA. It turns out many viral mRNAs look like this as well, but there are some differences. A typical mRNA has a 5' cap structure. They're usually untranslated regions at the 5 and the 3' end. So these are not translated into protein. They have other roles. They have regulatory roles. And then there's a poly A tail. The cap at the 5' end we talked briefly about during transcription. Here's the structure of a cap here on the left. It is basically a G or a guanosine residue linked by a 5 to 5' linkage to the first base in the mRNA. So there's base 1, and it's linked to base 2 by a 3 to 5' linkage. But the cap is reversed. Okay, that's why we call it a cap group. And this is the first base is a G, and it's usually methylated at this position, 7-methylguanosine, and there may be other methylations as well. And the methylation is part of the function of the cap group. This cap is important for translational efficiency, as we'll see today, stability of mRNAs, and a variety of other processes as well. Not all viral mRNAs are capped, as we'll see, and we'll see how they get around that. The untranslated regions, as I've said, have roles in stability of mRNAs as well. And then, of course, the mRNA has an open reading frame that directs the synthesis of a protein. So that's your typical mRNA. Let's look at it a little more closely. The 5' and 3' untranslated regions can vary in length. 5' typically 50 to 70 nucleotides, but there are some longer ones. And it often has secondary structures, stem loops and bulges and so forth, which we talked about before. And that secondary structure has a variety of roles. One of them is to regulate the efficiency of translation. So the more secondary structure in the 5' UTR of a message, the harder it is for the ribosomes to get through it. So if you were designing a message, let's say you want to make a protein in a cell because you want to sell it or use it for therapeutic purposes. You just want to make a lot of protein. You would use a short 5' untranslated region with no secondary structure. That's one way to get very efficient translation. You can't have no 5' untranslated region because, as you'll see, if you do that, then the ribosomes don't have room to sit down. So you have to have one of about 50 nucleotides and then no secondary structure. When there is a lot of secondary structure, the ribosomes can get through a certain amount because, as we will see, there is an unwinding activity associated with translation that can deal with that. But beyond a certain point, beyond a certain stability of the structure, the ribosomes can't get through. 3' untranslated region can also regulate translation. It can regulate the stability of the mRNA. And of course, the poly A tail is absolutely necessary for efficient translation. The machinery consists of ribosomes, of course. We're talking about eukaryotic cells, so these are ADS ribosomes composed of 40S and 60S subunits. And of course, ribosomes are mixtures of proteins and RNA. And the RNA has the catalytic activity. Protein is more of a scaffold. We think this is a leftover from the RNA world when protein synthesis began to evolve. Transfer RNAs, which attach the right amino acid to each codon, and then a variety of other proteins that function in the distinct phases of translation. We have initiation proteins, elongation, and termination proteins, and these are called IFs, EFs, and RFs, with an E in front of them to signify that we're talking about eukaryotic proteins. The way protein synthesis works in our cells is that the ribosome is brought to the mRNA through interactions with the cap. So the cap is absolutely important for translation. So here's an mRNA at the bottom. There's your cap structure. And it is interacting with a protein called EIF4E, which we also call the cap binding protein. And EIF4E in turn interacts with the red protein here called EIF4G. And you can see there are a lot of other proteins stuck onto EIF4G, including EIF3, and that in turn binds the 40S subunit. So that's how the 40S is brought to the mRNA. 40S binds 3, 3 binds G, and G binds E, and E binds the cap. And this is the series of these reactions are shown above here. But the essential point is that to get the 40S to the RNA, the cap is needed. There are other proteins here that are important as well. You see there's what's called a ternary complex, which consists of the initiator tRNA, which is a methionine tRNA, and a protein called EIF2, which we will talk about later, because this is a key point of regulation. And of course, the initiator is needed to start translation at that first AUG. This other protein here, EIF4A, is an RNA helicase. It unwinds secondary structure. This is what allows the ribosome to pass through the 5' non-coding region. So key point here is that the 40S subunit is recruited to the message via this series of interactions with all of these different proteins. Let's take a closer look at that bringing of the 40S subunit to the mRNA. So this red protein now is a little more visible here. We've taken away some of the others. EIF4G. And this you can think of as a scaffolding protein. Its protein sequence is shown as a line at the top. It's a very large protein, 220,000 Daltons in size. And this was discovered in cells because, as we'll see later, a virus cleaves it to shut off host cell translation. We didn't know anything about this protein until it was discovered in virus-infected cells. And that protein is a scaffold because it binds EIF4E, which is the cap-binding protein, and it binds EIF3, which in turn binds the 40S subunit. And you can see the binding sites for all of these proteins on this linear diagram at the top. There's the EIF4E binding site, EIF3. There's a binding site for EIF4A, which is the helicase, again, that unwinds the RNA, and a few other sites, which we don't have to worry about at the moment. One that's important, though, is this poly-A binding protein binding site, which is this little groove here at the end terminus of the protein. We'll talk about that in a moment. So this is how the 40S subunit is brought to the mRNA that has a cap. Now, we actually think that when mRNAs are translated efficiently, they're actually in a circular form like shown at the bottom here. We call this juxtaposition of mRNA ends. As I just told you, there's a binding site for poly-A binding protein here at the beginning of EIF4G. Poly-A binding protein is a cellular protein that binds poly-A. And so these interactions, PABP with EIF4G, PABP with poly-A, effectively circularizes the RNA. And we think this is needed. There's good genetic and biochemical evidence that this happens in cells, and this is needed for efficient translation, probably so that when ribosomes are finished translating an mRNA, they can just travel around on the mRNA and start over again at the beginning. All right, so this is what typical eukaryotic translation looks like. So now we return to the more complex view at the top. We have a whole bunch of proteins bound to the mRNA. We have a 40S subunit, and the 4E is bound to the cap. Now it has to find the start of translation. And this is a process called scanning, wherein the initiation complex moves along the messenger RNA until it encounters the initiation codon. And if there is secondary structure in the 5' UTR, like the one I've drawn here in green, it will be unwound by EIF4A, the helicase that's moving along with the 40S subunit. We don't know what happens to the 4E cap interaction. It might remain, in which case the RNA could be threaded through this complex, or it might dissociate. We actually don't know what's happening there. So the helicase that unwinds the RNA requires ATP, so that's shown here. This whole complex moves down the untranslated region. It encounters an AUG codon, which is the right one for initiation. Now, what does the right one mean? This is a complicated area that we don't have a lot of time to go into, but it's often the first AUG in a 5' untranslated region, but not always. There are exceptions. And secondly, there's sequences around the AUG that specify a good initiation context, as we say. So somehow this scanning initiation protein complex can detect all of that. And a lot of these other proteins here are able to detect the AUG codon. For example, EIF1, its job is to find the AUG codon. When the AUG is encountered, a lot of these initiation proteins come off. The 60S subunit then binds. So the 60S has been hanging around so far. It's not participating in any of this scanning. Until the AUG is reached, 60S comes on. Now we have the met tRNA there because that's been brought down with the initiation complex. And we are ready to make proteins. Okay? That's how it's done in eukaryotic cells. And as you will see, many viruses do that as well, but not all of them. All right, so the first question is, which of these statements is wrong about the 5' cap? One, it's a 7-methylguanosine joined to the second base of the mRNA by an unusual phosphodiester linkage. 2, it is present on most cellular mRNAs. 3, it is required for efficient translation by 5' end-dependent initiation. 4, it binds the cap binding protein EIF4E. 5, it is found on mRNA, but not pre-mRNA. 65% of you correctly answered it's found on mRNA, but not pre-mRNA. The cap, if you remember from our discussion of transcription, the cap is added after the RNA polymerase has begun transcribing 20 or 30 bases. So it's in the nucleus, and the cap is there in the pre-mRNA in the nucleus. So caps are there as well as mRNA, so that's the incorrect statement. All the other ones are correct. A lot of you picked A, but it is joined to the second nucleotide by an unusual 5' to 5' phosphodiester linkage. I want to now focus on what viruses do. Well, they don't do. Let's put it another way. How viral infection alters the translational environment so that viral mRNAs are favored over cells. And the first is we're going to talk about other ways to do basic translation that have been found in virus-infected cells that differ slightly from what I've just told you. The first one is a process called ribosome shunting. So I've just told you that if we have a messenger RNA like the one on A at the top, the ribosome will bind the cap through all these protein interactions. It'll scan down through the 5' UTR and find the AUG. And if you have a little bit of secondary structure in this 5' UTR, it's not a problem for the ribosome, as long as it doesn't exceed this number, negative 80 kilocalories per mole. That's a measure of the stability of the double-stranded RNA in the stem. Now, this is an experiment where a protein has been linked to this 5' non-coding region, and this RNA is translated in cells, and then the products are run out on a gel and probed by Western blot. So you can see the wild-type RNA encodes 2 proteins here that are made nicely. Now, if you put a stable stem loop structure at the 5' end here, and that's shown in red, this is very stable. It's negative 80 kcal per mole. Ribosome can't get through it. And you can see this mRNA is not translated. That's the third lane on the right, 5' insertion. And that's totally consistent with what I've told you. Ribosomes are moving along the 5' end. They bump into this. Bam, they can't go any further. The helicase gives up, and no protein is made. But look at when we put a stem loop, the same stem loop, at the 3' end of the UTR, just before the AUG. Yeah, that's fine. Protein is made. So apparently, ribosomes can get through it. How is that possible? They can't get through one that's placed upstream. Well, the answer is ribosome shunting. So the ribosomes don't always move linearly. They can bypass sequences, and that's shown on this slide. So here we have our secondary structure. Our ribosomes attached to a cap, not shown here. They can scan, and then they reach a specific structure which is present in RNAs in which shunting occurs, and they can bypass a lot of the secondary structure and make it to the AUG. So in our previous example, the ribosomes scanned up to a certain point. Let's take a look at that. So the ribosomes scan through this secondary structure and then are delivered to the AUG without further scanning. So the presence of that first stem loop is bad because it's interfering with scanning. But the second one doesn't matter because here's the structure that promotes shunting. The ribosomes recognize it and bypass that structure. And how it happens is kind of schematically shown here. You see here's this single stem loop is a summary of all the secondary structure in these 5' UTRs. And somehow the ribosomes can get across them without having to scan through all of them. So that's how they bypass that stable stem loop that we put in. So this has been found to happen in a number of viral mRNAs that are shown in the upper right. The plant pararetroviruses, those are those cauliflower mosaic viruses that have reverse transcriptase we talked about last time, adenoviruses, and a virus related to measles virus. They all do shunting. And this has been found in cellular messages as well. So discovered in viruses, now we know that cells do it. Other viruses don't have caps, like the picornaviruses, poliovirus, rhinovirus. Here's the poliovirus genome, which we've seen before. It's a 7.5 kilobase plus-stranded mRNA. And of course, when this gets into cells, it's immediately translated into a protein. This mRNA does not have a cap at the 5' end. It is polyadenylated at the 3' end. The 5' end has a little protein attached to it, VPG. And remember, that is the primer for RNA synthesis. So VPG-UU is used as a primer, and the protein ends up being on the RNA. You don't have a cap. How do you attract ribosomes? Well, the answer is that there's a highly structured 5' non-coding region which attracts them. So the ribosomes do not bind to the 5' end. There's no cap. They actually bind internally in this sequence, which I'll show you in a minute. So the 5' untranslated region of this virus, these family of viruses, quite long. It's 700 to 800 bases or so. And it has lots of AUG codons in it. And that, together with the presence of a protein, the lack of a cap at the 5' end, made people say, well, there's got to be something different about this mRNA. What is it? And so that was figured out by the experiment in this slide. And what was done here was to make a series of artificial mRNAs that could be used to probe how this 5' end of the viral RNA works. So here on the upper left is an mRNA. It's got a cap. It has poly-A tail at the 3' end. And it has two genes. Here they're called TK and CAT. But their function is so that we can measure something, much like the experiment I just showed you for shunting, where we're measuring protein synthesis by a Western blot. That's the function of these two. Now, if you have a mRNA with two open reading frames like this one and just some random sequence in between, what happens is ribosomes will bind the cap. They will translate that first open reading frame quite well. And then the ribosomes fall off when they reach the termination codon. A few 40S subunits keep scanning, and you might get a little bit of the second open reading frame, but in general, that doesn't happen very often. Now, if you take the 5' non-coding region of poliovirus RNA and you stick it in between these two open reading frames, so I'm showing it to you as a highly structured RNA region, now you get efficient translation of both open reading frames, as shown by the same number of proteins. So apparently, ribosomes can not only bind at the 5' cap on this, but also they can bind internally and initiate at the AUG of this second protein. So this was called an internal ribosome entry site, or IRES. And the experiment on the bottom actually provides even more evidence that this is a real phenomenon, because in this experiment, the translation of these two mRNAs is done in cells infected by poliovirus. And as we'll see at the end of today's talk, poliovirus infection leads to cleavage of EIF4G. And because of that, capped messages cannot be translated. So on the lower left, this mRNA, which just has some random sequence in between the two open reading frames, all right? You can't make any protein because the ribosomes cannot enter at the 5' end. They can't make the first protein, nor can they make the second. And on the right, when we have our internal ribosome entry site present, there's no first protein made. Again, ribosomes cannot access the cap because the cap binding protein has been inactivated. But you can make the second protein. So ribosomes clearly bind internally, and they can make that second protein. So this was a totally unique way of ribosome initiation. It bypasses the cap. Of course, it evolved because the virus doesn't have a cap on the 5' end. So another very nice experiment that was done a while later to prove this. So a corollary of this finding, this internal ribosome entry site, is that you don't need a free 5' end. So cap-dependent translation requires that free 5' end with the cap. But if you don't have a free 5' end, if you say circularize the mRNA, you can't get translation unless you have an internal ribosome entry site. So after a number of years after this experiment was done, people figured out how to circularize RNAs. It wasn't easy to do. And then they could do this experiment. So we have two mRNAs that have been circularized. They both encode a protein. The open reading frame is in yellow. The RNA on the right doesn't have an internal ribosome entry site, and if you put this in cells, it is not translated because there's no way for the ribosomes to attach. They need a cap, unless there's an iris shown on the left. You put an iris on this circle, the ribosomes can bind internally, and then they can access the AUG and make the protein. So this is a real phenomenon in not only virus-infected cells. Many viruses have now been shown to have internal ribosome entry sites, and they're present in cellular mRNAs as well. And they're highly useful in research. If you want to make one mRNA that encodes two proteins, you put an iris in between them, and you get efficient translation of both. And this is used very, very often, and one of the viral irises that's used is related to poliovirus. So what do these sequences look like? I've already given you a hint that they're highly structured, and in fact, these are 4 different kinds of viral irises. We have encephalomyocarditis, which is a picornavirus on the lower left. The polio iris is on the upper left. The upper right is hepatitis C virus iris. And the lower right is an iris from an insect picornavirus called cricket paralysis virus. So you can see they're all highly structured. They have no sequence homology, with few exceptions. There are some little areas like a pyrimidine-rich region, which is in common to all of them. But you cannot look at a sequence of a 5' untranslated region and say, aha, that's an iris, because there's no consensus sequence for an iris. What is important for the function of these structures is the secondary structure. Apparently, the secondary structure can attract the initiation machinery, either the ribosome or the initiation proteins, bring the ribosome right in near the AUG. So, for example, in the EMCV iris, shown here, here's the very 5' end of the viral RNA. There's the initiating AUG. EIF4G binds right here, very close to the AUG. So it brings in the ribosome, and then the ribosome scans over to that first AUG. So it's basically a substitute for the cap mechanism of initiation. So let's compare that now. On the top is the 5' end dependent initiation that we've talked about. That happens on cellular mRNAs, right? The 40S is brought to the mRNA through interaction of 4E with the cap, right? The iris containing RNAs of viruses in cells don't have caps. Let me correct that. The iris containing RNAs of viruses do not have caps, and they have instead this highly structured RNA, which in some cases binds EIF4G directly. And once you've got 4G bound, then you can bring in the 40S subunit through EIF3. There's another example of a viral iris which is quite interesting, because apparently for this iris, which is of hepatitis C virus, it's highly structured again. This can bind the 40S subunit directly. It doesn't need EIF4G. So this may be a very ancient virus, in fact. So remember, we had an RNA world at one time, and the RNA viruses evolved from them. And at some point, protein synthesis had to evolve. And this may be an ancient way of getting ribosomes to attach to mRNAs. So the requirements for these various initiation proteins vary here. Of course, for cap-dependent initiation or 5' end-dependent, we need all the initiation proteins. And we need all of them except EIF4E for this kind of particular virus. And then we only need a few of them for the hepatitis C virus. Now, look at the 4G in this middle panel here. You see I've cut off the 5' end. And we'll see that later on. Again, this happens in cells infected with some virus. EIF4G is cleaved. And that prevents 4E from bringing in EIF4G. So this turns off cap-dependent or 5' dependent translation. We'll look at that in a few moments. All right, the next question is what do ribosome shunting and internal ribosome initiation have in common? 1, cap recruitment of the 4DS subunit. 2, both involve RNA secondary structures. 3, ribosome scanning through the entire 5' UTR. 4, both require cap binding protein EIF4E. And 5, all of the above. Both involve RNA secondary structures. That's correct. 66% of you said that. That's in common to both of them. They both involve RNA secondary structure. The cap recruitment only applies to shunting. Doesn't apply to the iris. There's no cap involved, right? Cap-independent translation. Ribosome scanning, no one picked. And a few of you both require cap-binding protein. That's not correct because, again, it's not cap recruitment for the iris. All right, so we've now looked at 2 general initiation procedures, one involving a cap recruiting the ribosome, the other involving an iris, a secondary structure recruiting the ribosome, which does not require a cap. Both of these end up with this situation shown here, which is now the ADS ribosome is attached at the initiation codon, shown here as an AUG, and we have the MET tRNA there, and we're going to start translating. For most mRNAs, the initiator is actually an AUG codon. But again, through viruses, we've discovered that it doesn't always have to be. You know, the dogma has always been you initiate at an AUG, and when we sequence the human genome and we say, you know, there are 20 or 30,000 proteins encoded in our genome, that's because you're looking at open reading frames that begin with an AUG. But it turns out there's initiation at other codons as well. And these, of course, were discovered in viruses. And they're pretty neat. The one at the top is found in a virus called cricket paralysis virus. It's a picornavirus that paralyzes crickets. And it's actually being considered to be used in insect control to get crickets off of your crops. And the RNA of this virus, shown here, it's a plus-stranded RNA virus. It's shown here binding the ADS ribosome. The RNA folds in such a way to mimic an initiator tRNA. So the initiation by this RNA doesn't require the MET tRNA initiator because its RNA fools the ribosome into thinking, yeah, there's a tRNA here, and you can initiate translation at a non-AUG. So this RNA, you can see, is folded onto itself. This is the tRNA mimicking part in the P site. And this is base pairing with the first codon in the mRNA. And the next amino acid that will be put in, or the first amino acid will be put in, will be here an alanine, which is encoded by GCU. So this is methionine-independent initiation because the viral RNA is fooling the ribosome into thinking there's a methionine there. And on the bottom is another example of a viral RNA. This happens to be a plant virus RNA, where, again, you get non-AUG initiation, this time because the 3' end of the viral RNA, so this happens to be a capped plus-stranded RNA, the 3' end folds to look like an RNA, a tRNA. And in fact, it has a valine stuck onto it. The aminoacyl tRNA synthetases. Those are the enzymes in the cell that attach amino acids to tRNAs. They see this viral RNA and they say, oh, it's a valine tRNA, and they put a valine on it. So this translation begins with a valine and not a methionine. So these are two examples of how you can get methionine-independent initiation. And my question is, which has not been answered by anyone, how many of these do we have in our genome? Maybe we're underestimating the number of proteins that our genome encodes because we're not looking for non-AUG open reading frames. So let's look at this cricket paralysis virus a little bit, because it does an interesting thing besides non-AUG initiation, which I just showed you about here on the right. Here on the left is the genome of this virus. So this is a picornavirus. It's like poliovirus. It has a plus-stranded RNA genome, but unlike all the other picorna genomes, this RNA has two open reading frames. So it has an iris at the 5' end because it has VPG at the 5' end, no cap, so it has an iris to initiate translation of open reading frame 1. But then that ends in a terminator, and then there's a second open reading frame that continues beyond it. And there's a poly A tail at the 3' end. That second open reading frame is preceded by a second internal ribosome entry site. So ribosomes can bind internally to translate that. And that's where this non-AUG initiation occurs at the iris between the two open reading frames. And again, this is a cool iris because it can bind the ribosome like the hep C iris without any other initiation proteins. This is the only example I know of a eukaryotic polycistronic messenger RNA. That is an mRNA that has two open reading frames on it. In theory, there should be tons of these in cells because we know there are irises in cells. We have shown that functionally, but we don't see any examples of that. We only see one example in a family of viruses, and I'm not sure why that is. Here's the issue. In mammalian cells, eukaryotic cells, the mRNAs are typically monocistronic, means they code for one protein. One mRNA, one protein. We've already talked about the structure of that mRNA. That is in contrast to bacterial and archaeal mRNAs, which can be polycistronic. They can have multiple open reading frames on a single mRNA, so you can make multiple proteins from one message. And the reason that happens is because around the AUG on these bacterial mRNAs, there's a Shine-Delgarno sequence, which is complementary to one of the ribosomal RNAs. So the ribosomal RNA recognizes the sequence around the AUG, hybridizes to it, and it can initiate translation. So that can happen internally. But apparently, on most eukaryotic mRNAs, the ribosome can only enter at the 5' end via the cap and scan, and then once that first stop codon is reached, the ribosomes pretty much fall off, and you can't make additional proteins. I don't know why irises have not evolved to make polycistronic mRNAs in eukaryotic cells. They're certainly there, but we don't see them. We see them in viruses, and we see some other ways of getting around this issue, but no true polycistronic mRNAs. Now, another way that virus infection alters the translation landscape is in an attempt to make a lot of proteins from small genomes. But many of the viruses we've talked about have relatively small genomes. They don't encode a lot of proteins, or they can't. So viruses have evolved to try and maximize that coding capacity. And these are a bunch of ways that this can be done. We'll talk just about a few of these. And these are the examples with viral families on the right. So for example, alternative splicing, the fourth one. I've already talked about how alternative splicing of a message can give rise to different proteins from a single mRNA. And certainly viruses do that. This happens in cells, of course, as well. Some viruses make polyproteins. They make a long protein and chop it up. That's one way to get more proteins. You can make subgenomic mRNAs. We've talked about that already. You can have segmented genomes. So instead of having a long RNA, you can put it in pieces, so each piece encodes a protein, like influenza virus or reovirus. So think of this in terms of strategies for making a lot of proteins from one genome or one messenger RNA. Splicing we talked about. Internal initiation. I showed you that example of the insect virus where you have an iris in the middle of the genome so you can make two separate proteins. Some viruses, they actually have sequences in the RNA that allow viruses to keep translating past the stop codon. So I told you that whenever a ribosome in the cell encounters a stop codon, it typically falls off, so it can't reinitiate downstream. Well, viral RNAs apparently have sequences that keep the ribosomal subunits stuck on to the mRNA so they can translate a second open reading frame. That's reinitiation of translation. We have suppression of termination, where a stop codon can be suppressed with a tRNA, and finally ribosomal frameshifting. So all of these have evolved in viruses, again, to make maximal use of the coding capacity of the genome. So let's look at a couple of them in some detail. So first we have a polyprotein. Here's the problem. You have one RNA in a virus particle, say. It could be plus or minus, it doesn't matter. How do you get multiple proteins out of that? Because you can't make multiple messages. You can't make multiple proteins from a single mRNA in a eukaryotic cell. They're monocistronic. So a virus like poliovirus, which has a genome of mRNA, how would you make multiple proteins? Well, an easy way is to make a polyprotein. You make a long protein from this long open reading frame, and you chop it up with enzymes, proteases. And that's what these viruses do. Here's a polyprotein made by translation, and then there are two proteases embedded in the polyprotein. They're called 2A and 3C, and they make all these cleavages at the red and the blue triangles to give you the viral proteins. Flaviviruses do a similar thing. Their genome is shown on the bottom. Again, a plus-stranded RNA. They make a long polyprotein, which is then cleaved by proteases. For the flavies, they use a combination of viral proteases and cellular proteases to do this. So it's a very simple solution. It's not the only one. It's one way that you can make many proteins and get around this monocistronic limitation. Leaky scanning is another way. This is an mRNA from a paramyxovirus. It's a measles or a Sendai virus, mumps virus, if you will. And this virus has a negative-stranded RNA, a single long negative-strand RNA. It makes multiple subgenomic mRNAs from that. And from each mRNA, you can typically make a protein. But this virus has evolved to use leaky scanning to make multiple proteins from one RNA. So here is the schematic of the mRNA at the top. And there are two groups of proteins that are encoded, the C proteins and the P protein. And what we're doing on the bottom is expanding this very left end of the mRNA. So it's a capped mRNA. Nothing special there. Ribosomes bind and they start to scan down. And then at nucleotide 81, they encounter an ACG. Not an AUG. This is an ACG. But ACGs can be recognized by the methionine initiator. There's a mismatch there, but apparently that's okay. It's not great. So maybe 5% of the ribosomes will make a protein there. So that gives you this protein called C prime. It's 215 amino acids. But again, most of the ribosomes keep scanning. This is an ACG. This is not great. If you want to translate there, you can do that, but I'm moving on. So they keep scanning. They reach AUG at 104. So this is nice. This is an AUG. But the surrounding sequences are not good. I told you earlier that the sequences around an AUG can control the efficiency of initiation. So again, some of the ribosomes initiate there. They make this P protein, which is 568 amino acids. That's now in a different reading frame, so it's a totally different protein. So far we have two proteins from one mRNA. The ribosomes keep scanning, and finally they encounter an AUG at 114, which is in a really nice context. So they all initiate translation there, or most of them. So we have three different proteins. C prime and C are similar, but they have different N termini, and that can mediate different functions. We then have two other proteins, again in the same reading frame as the C, but lacking N-terminal sequences, at an AUG at 183 and 201. So these are quite far away. And these, in fact, involve ribosome shunting. So the ribosomes shunt from the 5' UTR over all these other AUGs and initiate either at the Y1 or the Y2. So a fraction of the ribosomes do that instead of engaging the others. So you have 1, 2, 3, 4, 5 different proteins made from this single mRNA. And there are more here that I haven't shown you. there are other ways of getting two different proteins in addition to this from this single mRNA. So it's really an illustration of what you can do by playing with the rules of initiation. Another way you can expand coding capacity is to utilize suppressors. So as you know, when translation machinery reaches a stop codon, here's UAA, one of the three stop codons. It is not typically recognized by a tRNA. There isn't a particular tRNA that would match it. But they're recognized by the termination proteins, which consist of ERF1 and ERF3. ERF1 is actually a protein that looks like a tRNA, and it binds with high affinity into the terminator site. And so what happens is the protein synthesis ends. We have here the growing polypeptide chain. When the terminator sits into the AUG, the stop codon site, that catalyzes the release of the peptide from that last tRNA, and protein synthesis is ended. So that's typically the way translation termination works. In very rare instances, you can have insertion of an amino acid at these termination sites. So for example, sometimes the terminators are misread by a normal tRNA, and you put some other amino acid here. So protein synthesis isn't 100% accurate, and sometimes you put the wrong tRNA in, you make an altered protein. In the normal course of biochemistry, those are probably going to be degraded by the check system of the host cell. But in this case, if you put an amino acid at this stop codon, you can make a longer protein until you encounter the next termination codon. So that happens. And it also turns out there are a few suppressor tRNAs in the cells. That is, very rare tRNAs that encode unusual amino acids like this one. Selenocysteine is attached to a tRNA that will recognize a UGA codon. So you have either misreading or a specific tRNA that recognizes the stop codon, and that can give you a longer protein with a different function. So viruses have evolved to utilize this. And here are two examples of that. On the left is what happens in a retroviral mRNA. So you may remember from last time, retroviruses make a genomic mRNA. They make a spliced form. The genomic mRNA is needed to get production of the structural proteins, which are GAG proteins, and the polymerase, the Pol region, reverse transcriptase, integrase, RNase H. The problem is that the open reading frames between GAG and Pol are separated by a stop codon. So here the GAG open reading. This is a capped message, by the way. So ribosomes come down, they translate GAG, and then they encounter this UAG, which is a stop codon, and they terminate and fall off. But the virus needs polymerase. It absolutely needs it. This is reverse transcriptase. So what happens is there's at a certain frequency of the time, maybe 5% or so, this suppression of this UAG codon occurs. And that suppression absolutely depends on the presence of a pseudoknot just downstream. Remember, a pseudoknot is a stem loop where the loop region is base pairing with a downstream sequence. So apparently what happens here is the ribosomes bump into this pseudoknot, and they pause just long enough so that a suppressor, a tRNA can come in and misread the stop codon. And then you can get, now you get a fusion protein made of GAG-Pol, and you get enough polymerase to suffice. Remember, polymerase is an enzyme, so you don't need a lot of it. So 5% suppression is apparently enough. If you get rid of this pseudoknot by mutagenesis, you no longer get suppression. So that's absolutely essential for that. On the right is an example of another family of plus-stranded viruses, the alpha viruses. And these have plus-stranded RNA genomes that are capped. And they also have two open reading frames separated by a stop codon. So the ribosomes come onto the 5' cap. They translate this first open reading frame, and then there's a termination codon there. But the second part is a structural open reading frame, which can't be accessed. Again, suppression of that at about 5 to 10% of the time gives rise to the structural protein. So a number of viruses employ this suppression to expand the coding capacity of their genomes. Another interesting mechanism is called ribosomal frameshifting. And this is observed actually throughout life as well, not only in viruses, but in cells and also in bacteria. And this is another example of how retroviral mRNAs are manipulated to access the polymerase coding region. I just showed you an example of how a suppression of a stop codon allows you to translate the polymerase from the retroviral mRNA. Other retroviruses do that in a different way. They do frame shifting. So the GAG protein is made by translation at the first AUG. There's a termination codon at the end, and that gives you the GAG protein right there. But of course, you need polymerase. And what happens in this case is that a certain percent of the time, the ribosomes back up one base and start translating again. And now they're in a frame that is not terminated at that point. That's why we call it frame shifting. The ribosome shifts its frame. And as you might guess, there are sequences in the viral mRNA that direct that frame shifting, just like there are sequences in the RNA that direct suppression, that stem loop structure. So ribosomes go down, they hit the terminator. Most of them fall off, but a few of them back up one. They remain bound. They back up one base. You're now in a different frame, and you can then translate the polymerase, and then you get a fusion protein of GAG-POL, which subsequently gets cleaved by a protease. Let's see how this works mechanistically. There are actually a couple of ways to do this, but this is one of them that will illustrate how this is working. When this frame shifting occurs, it usually happens around what's called a slippery sequence. And this is a stretch of A's and U's where if the tRNA backs up or if the ribosome backs up one, it's not going to make a lot of difference in terms of base pairing. So let's look here. Here's the termination codon at the end of GAG, UAG. And we've got a tRNA with the growing polypeptide chain in the P site. Then we have in the A site the next amino acid. Here, in this case, is leucine. And normally you would terminate here, and that would be the end of the GAG protein. But a certain percent of the time, the tRNAs slip. So now they've slipped back one base. So here's the terminator, and now you can see there's the A unbase paired. And the way these tRNAs are base pairing, you can see it's not so bad. You've got UA. This is a misbase pairing, AA. But then you have AU, AU, and UU. You know, the third position can be misbase paired sometimes. So this doesn't really matter too much. And now what you've done is shifted the reading frame. Because then when protein synthesis resumes, the next codon is going to be AUA instead of UAG. And that encodes for an isoleucine. All right, so you move back -1, new frame, bypass the stop codon. Pretty clever. Again, discovered in viruses, happens in lots of other situations as well. All right, next question is, compared with a polycistronic mRNA, a monocistronic mRNA, 1, does not require an AUG start codon, 2, does not bind the 40S ribosomal subunit, 3, has only one open reading frame, 4, is only found in viruses and bacteria, 5, all of the above. 90% of you got the right answer, which is C, only one open reading frame. Some of you said only a monocistronic. Compared with polycistronic, a monocistronic is found only in viruses and bacteria. It's not true. And of course, all of the above is not correct as well. Now let's end up with a little discussion about how viruses regulate translation and how cells respond in turn, because this is an arms race. One does something and the other does something in return, then the other responds. And this is a chart which you're not meant to see. It's a table which shows the different ways translation can be regulated in virus-infected cells. And all of these are initiation and elongation and termination proteins that are targets of regulation. But I've color-coded them to emphasize how the preponderance of regulation occurs at the initiation step, which is in blue. So most of the mechanisms we know about occur there. There are a few at elongation and termination. In other words, you can modulate the efficiency of initiation, of elongation, of termination. We've talked a little bit about that already, to bias it for the virus. And the cell can then try and compensate for that. So we're going to mainly focus on initiation because most of the mechanisms we understand happen at that step. A key step for regulating initiation is at the formation of the ternary complex. Remember the ternary complex shown at the top here. It's the initiator mTRNA bound to EIF2, which is an initiation protein, and a molecule of GTP. That's why we call it ternary. There are three things in it. All right? And when the ribosome starts to move from the cap, it's got associated with it the ternary complex, and eventually that ternary complex will donate the mTRNA when the AUG is found and the ADS comes on. And when the anticodon of the mTRNA sees the AUG, this GTP is hydrolyzed, and eventually it has to be recycled so that it's GTP again. So it comes off the complex as a GDP-EIF2 complex. This then is bound by another protein, which is basically a guanine exchange protein called EIF2B. And that holds this complex in place while GTP is exchanged for GTP. And then that can bind another mTRNA and go through the cycle again. So this recycling of the GDP and GTP is absolutely essential for initiation. If you interfere with that, you stop initiation. So this is a key regulatory point in translation in cells. The way this is regulated is by phosphorylation of this EIF2 protein subunit. And there are a number of cellular protein kinases that can phosphorylate EIF2 in response to various stresses. For example, endoplasmic reticulum stress. If there are a lot of misfolded proteins in the ER, or if a virus is making a lot of glycoproteins in the ER, then this kinase called PERK will phosphorylate EIF2. Will phosphorylate EIF2, which is shown down here. And that, of course, requires ATP. Amino acid deprivation also ends up in phosphorylating EIF2 by a protein kinase called GCN2. So if you starve cells for amino acids, they don't want to continue making proteins. That doesn't make sense. There's no amino acids. So protein synthesis is shut down. And finally, the presence of double-stranded RNA, which is a signature of virus infection, activates a kinase called PKR, which also phosphorylates EIF2. So in a lot of stress situations, the cell shuts down translation basically to either, if there's no amino acids around, that makes perfect sense. If there are misfolded proteins in the ER, you want to wait until they can be fixed and refolded before you make more. And finally, if you've got a virus infection, you want to shut down translation because maybe that'll stop the virus from spreading to other cells. So phosphorylation of EIF2 essentially makes it bind irreversibly to EIF2B. So now this GDP can't be recycled out for GTP. You end up not having enough EIF2 GTP, and translation stops. So that's the mechanism, all right? You sequester EIF2B, and so you can no longer recycle, and so you don't have enough ternary complex. So phosphorylation of EIF2 by any of these protein kinases results in a cessation of protein synthesis. This is a very effective mechanism of shutting off translation. It's so effective that if you are a virus and you can't get around it, you don't exist anymore. Every virus that we know of that encounters these kinases in infected cells have a way to get around them, and I'm going to tell you about those. First, this protein kinase PKR. This is the one activated by double-stranded RNA. So double-stranded RNA is typically made in virus-infected cells. They're either intermediates of replication of RNA viruses, or for DNA viruses, you can transcribe both strands of a double-stranded DNA, and these RNAs will hybridize. You get double-stranded RNA. And that double-stranded RNA will activate the protein kinase. So PKR, as it's produced in the cell, is not active as a kinase. As soon as it sees double-stranded RNA, it gets activated, and then it can phosphorylate EIF2. How it's activated, you start with inactive PKR. They have a catalytic domain and a double-stranded RNA binding domain. Two molecules of PKR bind a double-stranded RNA. They phosphorylate each other, and then they are active, and they can phosphorylate EIF2. So double-stranded RNA allows the two molecules to bind, activate each other, which then shuts off translation. So PKR is actually induced by virus infection. When we talk about interferon later on, you will see that it's an interferon-stimulated gene, and it's activated by virus infection. So as it's produced initially, the protein is not active as a kinase. It has to be activated by double-stranded RNA. By the way, this protein and its phosphorylation of EIF2 also has a role in uninfected cells. There are situations where you want to regulate translation, and that is done by a cellular protein called PACT. So this happens independent of virus infection as well. So phosphorylation of EIF2 leads to inhibition of host translation. And of course, that's to stop virus infection. It also induces apoptosis, programmed cell death. The cell dies, the virus infection doesn't spread. This is a common defense against infection. So as I've said, every virus has a way to get around this. Otherwise, they're not here anymore. They can't replicate when there's no protein synthesis. And I just want to show you a couple of ways that viruses get around this. There are dozens, as you might expect. And this one I like. This is adenovirus. Adenovirus infects the cell. It makes double-stranded RNA because it's a double-stranded DNA virus that you get transcription of both strands. That RNA, double-stranded RNA, activates PKR early in infection. But then the virus makes a small RNA called VA-RNA-one. This is a Pol3 synthesized RNA. It is a highly structured RNA. It's shown here. Its structure was recently solved. And this RNA has the property of binding only one molecule of PKR. Only one. And so PKR can't activate another PKR. So it's never activated. EIF2 is never phosphorylated. The virus merrily goes along and makes its proteins. Okay, so this is the way it antagonizes PKR. Normally, the double-stranded RNA would lead to activation of PKR, phosphorylation of EIF2, and inhibition of protein synthesis. But this little RNA overcomes that by binding up all the PKR and preventing it from getting activated. And if you take out the gene for this VA-RNA from the virus can't replicate very well because it can't make protein. Protein synthesis is shut off. So that's a cool way of doing it. And I just want to show you some examples of other viruses. You just need to know in general that viruses have to antagonize this PKR phosphorylation system. You don't need to know individual mechanisms, but I want to show you here, different viruses are listed in the middle, and these are the targets at which they antagonize this phosphorylation of EIF2. So I just told you about adenovirus VA-RNA. Here it is, VA-RNA, which is an antagonist of PKR, right? It binds PKR and prevents it from activating. Lots of other viruses make similar RNAs and also proteins. They can also bind up double-stranded RNA. They have proteins that bind double-stranded RNA and prevent it from binding PKR. Some antagonists mimic EIF2 alpha. They're proteins made by the virus. They look exactly like EIF2 alpha. So all the PKRs phosphorylate that and they ignore EIF2 alpha. And then there are phosphatases that take the phosphate off. Just amazing array of ways to get around this because this is such a powerful antiviral mechanism. PKR is an interferon-induced enzyme that is activated by something leading to phosphorylation of something else and inhibition of translation. 84% of you pick B, which is it's activated by double-stranded RNA, and of course phosphorylates EIF2 alpha, not EIF2B. EIF2B is the GTP recycling protein that binds the phosphorylated EIF2 alpha. That's not right. Another way that cells respond to virus infection. So we talked about phosphorylation of EIF2. There are many other ways that cells respond. Another one I want to tell you about is by forming stress granules. All right. So let's say we have translation going on in the cell. It's diagrammed in the upper part by a circular RNA being translated. Let's say there's some kind of stress applied to the cell. It could be a virus infection, could be serum deprivation, amino acid starvation, high temperature. These are all stresses that cells respond to by stopping translation because it makes no sense to translate when the cell is under stress. Okay? So what they do is the translational complexes become stalled. The ribosomes stop. There are specific mechanisms for doing that. And then they're shunted into what are called stress granules. These are actually specific structures that form in the cytoplasm. They contain these mRNAs, stalled mRNAs with the ribosomes still on them, and they are built with a variety of cell proteins, which are shown here, like G38P, TIA1, and TIAR, and a bunch of others. These are rather recently discovered structures, and the idea is in a stress granule, there's no more translation. Translation is shut off, and maybe the cell will let it start again if things return to normal. Of course, if it's a transient condition like lack of amino acids or temperature, the idea is that the RNAs can come out again and resume. But if it's a virus infection, maybe you want to stop the virus right there so you don't do any translation. Now, viruses, of course, have evolved to get around this. They don't want their mRNAs put into a stress granule. They're not going to be translated. So a variety of strategies, which are diagrammed here, have evolved to prevent the formation of these stress granules. For example, poliovirus, one of the proteases of poliovirus cleaves G38P, or is that G3B? Should be B, G3BP. It's wrong, but it doesn't really matter. The point is this is an essential component of the stress granule, and when it's cleaved, the stress granule can't form. So that's how that virus gets around it. Other viruses, hepatitis C virus, actually steals G3BP to form the replication complexes on which it replicates its RNA, so stress granules don't form. And a variety of other viruses steal these complexes. This is a pretty neat one. The flaviviruses have double-stranded parts of their genomes that bind up G3BP and TIAR, and again, they prevent stress granules from forming. So it's another way where the cell is trying to stop translation because it's sensing that it's virus infected, and the virus is getting around it. It has evolved. If you don't have a way to get around it, you don't survive. So these mechanisms have evolved to get around this. Really interesting stuff. Last thing I want to mention is the role of microRNAs in translational regulation in a virus-infected cell context. Now, microRNAs, as you know, are small, non-coding RNAs. They can be encoded within our genome. We have over 1,000 of them in our genome. Some virus genomes encode them as well. They're transcribed as primary transcripts by either Pol2 or Pol3. That's done in the nucleus, and then they're processed to 22 nucleotide single-stranded microRNAs, which then bind to a target RNA and cause it to be either degraded or to have its translation inhibited. All right? These are an incredibly interesting area of research, of course. Nobel Prizes have been awarded already for microRNAs, and they regulate many of our genes, but it turns out that they can regulate virus infections as well. So this is how they work. They're, again, transcribed as primary microRNAs in the nucleus. They go through a series of processing steps, both in the nucleus and outside in the cytoplasm, and end up getting that 22 nucleotide microRNA, which is shown here in green. This microRNA has complementarity to typically a cellular gene, and when the RNA, the microRNA, binds the mRNA, it can do one of two things. If the base pairing is perfect, so if the microRNA matches the mRNA perfectly, that mRNA is then degraded. And there's a nuclease complex which contains, among other things, the Argonaute protein that leads to degradation of the mRNA. And of course, a degraded mRNA cannot be capped. But if the base pairing between the microRNA and the mRNA is not perfect, and there are examples where you have bulges and mismatches and so forth, then you have translational inhibition. You don't have degradation, but you have interference with ribosome binding at the 5' cap in the process that we talked about. So these are used to regulate cellular genes within us. What's the role in virus infection? So there are certainly microRNAs in the cell that target cellular genes that are needed for viral replication. There's a number of examples where a virus requires a certain cellular gene, and the cell can respond by turning it off using a microRNA-mediated mechanism so the virus cannot replicate. Viruses also have microRNAs within their genomes that regulate their own replication as well. For example, the herpes viruses, which set up latent infections in us, we'll talk about those in a couple of weeks, they regulate their gene expression to keep a latent infection by using microRNAs to silence lytic replication. Now, viruses can also utilize the cellular microRNAs to regulate their infections. And a good example is shown here. There's a microRNA, these are called MIR, and they're given a number. MIR-141 happens to be, it's a cellular microRNA that is complementary to a part of the mRNA that encodes EIF4E. Right? Remember, EIF4E is the cap binding protein. So we encode a microRNA that will regulate the production of 4E by the microRNA binding to 4E message. So you know, this exists in cells because we have to regulate translation, and this is one way to do it. Some viruses take advantage of that. So for example, enterovirus related to poliovirus induces the synthesis of MIR-141. And what does that do? That shuts off any translation that is dependent on a cap. And of course, this enterovirus doesn't have a cap. It's got a protein, it's got VPG, it has an iris, so it doesn't care about EIF4E. So it's basically shutting off host translation. So there are a number of examples where viral and cellular microRNAs play into replication. And one of the really interesting ones, which I'm not showing you today because it has nothing to do with translation, is that hepatitis C virus, a liver pathogen, has evolved to require one of our liver microRNAs called MIR-122 in order to replicate. Can't replicate without it. And an antiviral drug has just been designed and is in testing to block that microRNA, and that prevents the virus from replicating. So the last topic has to do with something which I've already mentioned, and that is viruses shutting down host translation. So I've already mentioned that an enterovirus upregulates MIR-141, which will shut down cap-dependent translation in the cell. That only works, of course, if a virus doesn't need a cap. There's another example where viruses shut off translation by cleaving EIF4G. Now remember, the goal here is to have the entire translational apparatus devoted to viral mRNAs. You don't want any competition for amino acids, any of the translation components. You want only viral mRNAs to be translated so you can make optimal numbers of virus particles. Here's an experiment where cells are infected with polio, and we're looking at the rate of protein synthesis with time after infection. And within 2 hours, the translation is shut off in poliovirus infected cells, and then it resumes, and what occurs is all virus-specific protein synthesis, whereas in unaffected cells, there's no change, of course. And on this protein gel on the right, you can see this at early times, at 0 time after infection. These are all cellular proteins that are being fractionated on this protein gel. And then with 1, 3, 5, and 7 hours after infection, you can see the cell proteins are replaced with these darker bands, which are all virus-specific proteins. So this happens by a number of interesting mechanisms. And one of them I've already mentioned, which is cleavage of EIF4G. So remember, 4G is a scaffolding protein that links up the cap binding protein with the ribosome. So coronaviruses encode a protease 2A, which has a primary role of cleaving the viral polyprotein, but it also cleaves EIF4G right there at the N-terminus. The effect is that 4E can no longer interact with 4G, so any message that's capped will not be translated. But of course, the coronaviruses, polioviruses, don't have a cap on their messages. They have VPG, and they use an iris for internal initiation, so they don't care about 4E. They do need 4G, and cleaving the N-terminus off apparently allows them to still use the rest of the protein to bring the ribosome in, because 4G binds the iris directly. There are also other ways to regulate translation in virus-infected cells. For example, EIF4E needs to be phosphorylated in order to optimally bind to caps. And in some virus-infected cells, the phosphate is taken off. and that makes 4E work very poorly to recognize the cap. And in viruses that don't require cap-dependent translation, this is an effective strategy to shut off the host. The 4E itself can be regulated by a protein called 4E binding protein. When 4E binding protein is phosphorylated, it cannot interact with EIF4E. But in some virus-infected cells, it's dephosphorylated. That 4E binding protein can now bind 4E and prevents it from interacting with 4G. And again, the net result is that cap-dependent translation is inhibited. So these are all very effective strategies to direct the translational machinery only to viral mRNAs. And there are only a couple out of all the dozens that are out there, because apparently it's very important if you're a parasite of host translation, you need to have that system dedicated to your own means.